No, este. De este lado. No, ese no.
Muy buenos días a todos. Hoy día tenemos el honor de contar con la presencia del doctor Michel Laurin. Voy a hacer una breve biografía de su carrera científica. Michel Laurin obtuvo su doctorado en la Universidad de Toronto en 1994 acerca del origen de los amniotas, es decir, de los reptiles y mamíferos. Hizo 3 postdoctorados en Berkeley, París y Berlín, antes de aceptar un puesto como científico investigador del CRNS en París, es decir, el Centro Nacional de Investigaciones Científicas de París. Ahora dirige el equipo de filogenia y diversificación de metazoarios, uno de los 3 mayores grupos de investigación en el mundo. Sus principales intereses de investigación incluyen la evolución de los tetrápodos paleozoicos, la datación del árbol de la vida mediante fósiles y la nomenclatura filogenética. Ha formado parte de los consejos de varias sociedades científicas, como la Asociación Paleontológica de Francia, actualmente vicepresidente, y la International Society for Phylogenetic Nomenclature, actualmente secretario, que desarrolla el Phylocode, es decir, el Código de Nomenclatura Filogenética a partir del año 2020. Tiene algunas responsabilidades editoriales en revistas científicas. Es editor en jefe de la Academia de Ciencias de París, del Contest Paleoworld, editor asociado de Zoología Scripta, Frontiers in Paleontology, Contributions to Zoology, etc. Ha publicado 184 artículos en revistas especializadas, 38 capítulos de libros y ha escrito 2 libros. Bueno, ahora Michel Laurin va a presentar una clase abierta para toda la universidad y para quien la quiera mirar en vivo, que se titula El origen, el paleoambiente, diversificación temprana de los tetrápodos. Muchas gracias, bienvenido.
Muchas gracias, Lara, muy amable. Lo siento, debo hablar en inglés porque mi español no es tan bueno. Disculpe. Y si hablo demasiado rápidamente, dime, porque naturalmente hablo rápidamente en francés y inglés. Okay, so this talk is about the origin and early diversification of tetrapods. Here's the outline. We will first study a little bit the diversity of early tetrapods and their predecessors. So we'll talk a bit about nomenclature, taxonomy, and old and recent phylogenies of the group. Then we'll spend more time studying the habitat of early stegocephalians. These are the limbed vertebrates, which are often called tetrapods. I will explain the difference in a minute. And we will study this habitat through the associated fauna. Which taxa are associated with these stegocephalians, and what does this tell us about their habitat? Then we will look into a few sedimentological arguments, briefly, because this is not my specialty. Recent isotopic data, also briefly, again, not my specialty, before concluding on the habitat of these early stegocephalians. I will then cover briefly the origin and early evolution of digits, the fingers, in the hands and feet. And then, last but not least, we will try to answer this very interesting but elusive question: why did our ancestors come out of water and onto land? The answer is not obvious. So, let's start with stegocephalian nomenclature. Throughout this talk, I will use phylogenetic nomenclature because it gives precise, unambiguous meanings to names, taxonomic names. And here on the screen, you can see already 2 names that I will use and what they mean. Lissamphibia, to the left in pink, is the smallest clade that includes all of the extant amphibians, the frogs, salamanders, and the gymnophionems, which are all endless amphibians. Amniota is the smallest group that includes mammals and reptiles. Birds are reptiles, so that includes all of them. So these are the clades in pink and green that you see highlighted now. Amphibia, and this is an important point here, amphibia, in this sense, in the phylogenetic sense, is only the largest clade that includes these amphibians, but not amniotes. It is the large clade that you see in pink on screen to the left. So amphibians are not the first tetrapods, for example. They are often called amphibians in literature, but in my opinion, this is an error, and this is misleading, as we will see on the section on paleoenvironment. Reptilomorpha, also called Amniota, is the largest clade that includes amniotes, but not the amphibians. It is the large clade in green. Both of these together form Tetrapoda, which is also a crown group. It is delimited by, it is the smallest clade that includes Lissamphibia and Amniota. So in this sense, Tetrapoda does not include all of the limbed tetrapods. It includes all of the limbed tetrapods that live today. But in the past, there were limbed tetrapods that lived that belong outside of this group, right? Because normally when there is a synapomorphy for a group, you know that it appeared sometime earlier. You don't know how long earlier. But the fossil record clearly shows that the first limbed vertebrates were stem tetrapods. In that sense, they were not part of Tetrapoda per se. So the clade that includes all of the limbed tetrapods I will call Stegocephali, or the vernacular equivalent, Stegocephalians. And this talk will emphasize Stegocephalians, their origin, and the environment in which they lived, from the late Devonian until sometime in the Permian. So, now that we've covered nomenclature, let's start a little survey of the diversity that existed in the Carboniferous and the Permian. The first taxon I'd like to mention is the lepospondals, which are amphibians, very likely. These are known from the early Carboniferous to the late Permian. So they lived from about 330 to 260 million years ago, approximately. These were small animals, more or less in the same size range as extant amphibians. So most were less than 40 centimeters in total length, tail included. The giants of the group measured about 1.50 meters, but there are very few of these. They inhabited several habitats from aquatic to terrestrial, and they have often been considered polyphyletic in the past. That means that they might be constituted of several distant clades that are only distantly related. But these old hypotheses did not stand up to scrutiny. Many recent analyses suggest that they form a clade. And one of the apomorphies that demonstrates this is the vertebral structure. If you look at the bottom here, you see in blue the pleurodentrum. This is the main part of the vertebral centrum. This is where what surrounds the notochord in development. It is a single cylindrical element, and it's fused firmly to the neural arch. So this type of vertebrate is characteristic of lepospondals, but also lissamphibians and amniotes. We have similar vertebrae. And this is a derived condition because the first stegocephalians had vertebrae like these here to the left. These are called multipartite centra because what you see here in blue is a paired pleurodentrum. So you don't see that it's paired because you're looking at it here in left lateral view, but there's one left and one right element. It's like half of a crescent. It articulates with the neural arch. And ventrally, you had an intracentrum. It's crescentic, and this is highlighted here in red. So these were joined, of course, by cartilage and ligaments in live animals. But this is not a solid vertebral structure as the one to the right. So now let's look at another group of animals called temnospondals that have often been considered amphibians, but we'll come back to this because in my opinion they're not. More than 150 genera have been described. These are very diverse. In fact, a good proportion of the early stegocephalians are temnospondals. They come in all sizes, from about 10 centimeters in total body length to up to 6 meters for the giants. There were not many that measured more than 3 meters in length, and these were late taxa in the Triassic, for example, or the Jurassic. They are known from the early Carboniferous to the early Cretaceous, so this is a very long-lived group, so between 330 and only 100 million years. But there were almost no temnospondals left after the end Triassic crisis. there were very few reds. And they also inhabited several environments, aquatic, some were amphibious, and they were terrestrial. Here you see two of them, two temnosponds, and one of them, Ariops, is lodging to catch this little shark. We'll come back to this association, fairly interesting, later. Because here you can see the implications for paleoenvironments. You have what was called an amphibian, which is not, and a shark. Amphibians today are freshwater, and sharks are mostly marine. So this is weird. Now, according again to old phylogenies, the reptilomorphs, so this is the panamniotes, the taxa related to amniotes, included embolomeres, semoiomorphs, this is here a semoiomorph, dialectomorphs, like these animals, and these were the only anamniotic herbivores among land vertebrates at the time, and amniotes, like this Dimetrodon, a synapsid, which means a stem mammal. It's more closely related to us than to iguanas, for example, even though it doesn't look like it. So all of these are reptilomorphs, and they share a tabular parietal contact. So on the skull table here, you have this parietal in blue that shares a suture with the tabular, the bone in red. This is a derived condition because the primitive one, which you can see here in Ichthyostega to the left, this is a Devonian stegocephalian, is for these two bones to be separated by contact made by two other bones, the postparietal here and the supratemporal here. So this is one of the synapomorphies that was thought to unite all of these taxa, but I will show you this is not quite correct. We'll come back to it. So until a few years ago, we thought that almost all stegocephalians, that is the limbed vertebrates, were tetrapods. And this phylogeny, which is old but still accepted by several colleagues, is shown here. So we thought that these amphibians were temnospondals, that is, you see here the temnospondals are shown as a paraphyletic group that lead to these amphibians. Lepospondals were also thought to be amphibians, but more distantly related to those amphibians. Whereas embolomeres and semoiomorphs were considered to be reptilomorphs, as I just mentioned, along with the dialectomorphs and amniotes. These ideas stemmed from the works published by Cope in the 1880s, so that's what I mean by old phylogeny. It's been like more than 140 years since these ideas have been around. So perhaps because of this, it's difficult to convince people that this is wrong. I experienced this because I found out in my thesis, defended in 1994, that this doesn't hold. And I'm still trying to convince the community that the new phylogeny is correct. So what my analysis show is that temnospondals are stem tetrapods, placed here on the tree. Similarly, embolomeres and semoiomorphs are also stem tetrapods. They are not reptilomorphs, even though in probably 90% of the papers that have still been published in the last 10 years, they are being called reptilomorphs, but I think it's wrong. In fact, remember the parietal tabular contact that is supposed to unite embolomeres, semoiomorphs, and amniotes? In fact, it appeared more basally at this node below embolomeres, and it's characteristic of all this clade. In fact, if you look at the lepospondals, most of them have this feature also. And in some amphibians, you cannot see it because they lost these bones. And of course, they're present in amniotes. So this character is congruent with this new topology as well. So the amphibians in my phylogeny are descendants of lepospondals, and they are monophyletic, which is congruent with also molecular studies that suggest a relatively recent origin for this group in the Permian or Carboniferous. And most of the computer-assisted phylogenetic analyses that were published after mine obtained compatible results, but sometimes they excluded some taxa, so you couldn't realize this. For example, this is Carroll 1995. Its topology is perfectly congruent with mine, but it excluded these amphibians. So he could not see from his phylogeny if semoiomorphs, for example, were reptilomorphs or stem tetrapods. It's impossible to tell because you're missing the amphibians. It's a key piece of the puzzle. Similarly, Patton et al. 1999, same exclusion. Anderson 2001, he included one gymnophonon, and sure enough, it falls within the lepospondals where my topology predicts. Only a few phylogenies like Ruta and Coates 2007 or Anderson et al. 2008 obtained different results. But my former student, David Marjanovic, showed that if you correct these matrices to remove obvious contradictions between the scores in the matrices and the published descriptions, you obtain a phylogeny congruent with mine. And we still published a paper this year about this, and a big 200-page monograph in 2019. Still not enough to convince most colleagues, but one day we'll get there, I hope. Anyway, in this talk, I will consider that terminal are stem tetrapods, not amphibians. So, this brings us now to the second point in this talk. Where did our precursors live before they come out of the water? Did they live in fresh water, as comparisons with extant amphibians would suggest? As you know, when you taught introductory biology, they show you a frog and they say, well, it's something similar to the first tetrapod. I think this is wrong, but anyway, extant amphibians live in fresh water, as you can see, frogs, salamanders, and others. But perhaps our ancestors came out of the seas, and this is supported by the rich biodiversity that you can see on many coasts, most coasts in fact. It's very diverse. So, in 2010, I published a review in which I already concluded that our ancestors came out of the seas and that they retained the ability to tolerate salt water for most of their evolution. And in fact, if you look at this tree, you see that only less amphibians lack it. Because we amniotes, for example, can go into the sea. Most of you swim into the sea. Whales swim into the sea. Seals, turtles. Right? For amniotes, it's not a problem going to salt water. Only less amphibians had a problem. And still there are exceptions. So here was my conclusion, is that probably most early stegocephalians lived in partly salty water, that is brackish. They could probably also enter freshwater, but they were by no means excluded to freshwater only. And some probably lived in the seas also, in coastal environments mostly. But 14 years have passed. So does this conclusion still hold up? And what have the new data, well, what do they say about this? Do they reinforce my intuition or do they refute it? So that's what we'll look at now. Let's first look at our precursors, that is the finned vertebrates that were still primitively aquatic, before they had digits and before they moved onto land. Where did they live? Was it in the seas or was it in fresh water? And so a good place to start is the Old Red Sandstone, which is a series of deposits that range from Silurian to Mississippian, early Carboniferous in age, but the bulk of it is Devonian, and it includes many of the first vertebrates that were found. For a long time, they were among the first vertebrates. These have long been considered to be freshwater deposits. If you look at any paper more than 20 or 30 years old, you will almost certainly see that it's freshwater. And yet, it's probably wrong. But first, look at these nice pictures. You can see that some of these old red sandstone deposits make quite picturesque formations and structures. Anyway, this interpretation started being questioned when we found a few brachiopods, namely lingula, in these deposits. Now, this is not a new finding. Look, this is from Gougen Emig, 1985. So this is almost 40 years ago. And sure enough, these brachiopods, well, their shells are relatively fragile. They don't think that these were transported from a distance. These were autochthonous elements. So they indicate a fairly marine environment because brachiopods are not very tolerant of salinity tolerant fluctuations. They require fairly high salinity. This has been studied in extant taxa. And more recently, a recent isotopic study showed that there is a fairly strong marine influence among the vertebrates found in some of the old red deposits. I'm referring here to Godard et al. 2018. We'll come back to this paper later in the brief section on isotopic data. But they found that their conclusion was that these stegocephalians that they studied, including Ichthyostega and Acanthostega from East Greenland, lived in coastal brackish environments in which there was a mixing of seawater and freshwater. So all of this refutes the old hypothesis that all of the old red sandstone was freshwater deposits. Perhaps some of it is, but not all. Some was marine or coastal brackish. Also, if you look at the distribution, the geographic distribution of early stegocephalian in the Devonian, the latest Devonian, that's Stamenian, 360 million years ago, you see that this distribution is not compatible with strictly freshwater taxa. Look, most of these fossils occur in Euramerica, in this block, where they could, of course, have spread through rivers and lakes. But they also occur in Australia here, at the other end of this block. And what you see here, I should mention, The deep blue is deep oceanic basins. The light gray is epicontinental seas that you see here, and the stippling indicates emerged lands. So to go from here to there, you need to cross epicontinental seas. But more importantly, there is also now a stegocephalin from the North China block. It is separated from the others by a deep oceanic basin. There's no way a freshwater taxon could go from here to there. Impossible. So at least some of them were able to cross oceanic basins. Now the first vertebrates, including the first sarcopterygians, are clearly associated with marine fauna, and this is clear at least until the early Devonian. After that, the picture becomes less consensual, less obvious. But you can see here some of these taxa that are associated with early vertebrates, like the sea scorpions, the Eurypterids. We'll come back to these, they're very interesting. Placoderms, this is a sarcopterygian, so it's a tetrapodomorph, meaning that its closest relative today is tetrapods, even though it had fins and internal gills. But you see, this one lived close to crinoids. Sharks, crustaceans, anyway, marine fauna. But let's then move forward in time to the Promo-Carboniferous, which will be the bulk of this talk. In which habitat did most of these Promo-Carboniferous stegocephalins live? I suggest that they have often been considered freshwater, among other reasons, because they were called amphibians, and today's amphibians are freshwater animals. But this is a wrong reasoning because, as I showed you, the Thamus funnels are not really amphibians, they're stem tetrapods. So there's no reason to infer that the attributes of list amphibians are shared by these taxa. They are no closer to list amphibians than to us, and we tolerate salt. Also, these are often associated with sharks. So to determine their habitat, as best we can, we will look at the associated fauna. And we'll start with sharks. Now let's look at extant shark constrictions. 43 species, this is less than 4% of the 1,200 currently recognized species of constrictions venture into freshwater. I say venture, that does not mean they are restricted to freshwater. Many of these are marine taxa that go occasionally into freshwater. For example, Carcharodontis leucas. It is a widespread shark, you find it in many oceans and seas, but it also goes regularly into freshwater, and it even reproduces there. For example, this is a picture of Carcharodontis leucas, and it was found here in this paper, for which you see the reference, so a bit more than 100 kilometers away from the open sea. But you see, this is a broad estuary. Much of the water here is brackish, although where it was found may have been completely freshwater. This is an enlargement of this little rectangle that you can perhaps barely see. It's in northern Brazil. Okay. Anyway, but this is not the most freshwater adapted constricting. This record belongs to the Potamotrigonidae, the freshwater skates. These live all of their life. They complete the life cycle in freshwater, but they are also viviparous. No extant constriction lays eggs into freshwater. So I think that when you find a constriction egg capsule in a Paleozoic sediment, it means this was a marine environment, unless it was transported there for long distance. And this is interesting because we have such egg capsules from localities that have been interpreted as freshwater, which I think is wrong. Now, more precisely, the xenic antiforms are the so-called freshwater sharks, most frequently associated with these stegocephalians. And they often occur in sites where you don't have obviously marine fossils, that there are no coral reefs, echinoderms, and things like that, or ammonites. However, these are also known from marine sites where they are believed to be autochthonous. And there is such a description here, for example, Hampe and Ivanov 2007. So my conclusion is that these sharks probably inhabited several environments. They occurred probably in coastal environments, in brackish water also, and even in freshwater. But they cannot be used as freshwater indicators, contrary to what has been done with what has been done recently. They don't tell you, in fact, very much. And if anything, I think they probably indicate that the sea was closed, or the water was not necessarily salty. Moving on now to another interesting taxon, the Paleozoic Spirorbis, which I put in question mark. You'll see why in a second. This is a fairly common fossil, and it occurs in many fossiliferous localities that have yielded vertebrates, including Juggins, which is famous. It's a late Carboniferous locality in eastern Canada that has yielded the oldest known animals. It's about 319 million years old, according to the latest dating. The traditional interpretation of that locality is that it is an intramountainous freshwater basin, or freshwater locality. But this raises two problems. First, Spirorbis is an extant annelid. You wouldn't expect it to occur in the Carboniferous. Second, it's marine. What would it do in the freshwater locality? It doesn't make sense. And it doesn't because, in fact, as you may have guessed it, the Spirorbis is not really Spirorbis. It's not even an annelid. It is a spirobiform microconcave. This is an entirely different clade, not closely related at all. And in fact, all of the fossils that have been called Spirorbis before the Cretaceous are neither Spirorbis nor annelids. They are microconcaves, which appeared in the Ordovician and are documented all the way to the Jurassic. And this has been ascertained by the microstructure of the shells, which differs between microconcaves and annelids. They are not the same. Now, these are thought to be related to phoronids, which is also a marine taxon. These are extant phoronids. And there is a debate currently about the habitat of those macroconcaves. But this review that I documented here by Kierlowski, Kordesh, and Castle 2015 makes a very strong case that Spirorbis was a coastal marine form that inhabited also brackish water in estuaries and was sometimes perhaps carried by storms into relatively freshwater environments where they survived but did not thrive and did not make colonies. And that would explain pretty much all the occurrences we had. So whenever we find this, we have to think that at least the sea was not far. And where there are colonies, the water was probably salty. Let's now look at the horseshoe crabs, which are often associated fairly often with early Stegocephalians, the Xiphocerans. They survive today, of course. They are not very diverse, but these are marine and euryhaline forms. One of the species, only one out of maybe 6 or 8, something like this, ventures into fresh water, or some water that is nearly fresh, but never very far from the seas. The ecology of these taxa has not been studied as intensively as it deserves. See, one of the most interesting papers I found is the Annandale 1909, so it's more than 100 years old, but it remains relevant because I have not found similar studies in more recent ones. Anyway, here she said that Limulus rotundicoda, which is now considered Carcinoscopus, is mainly, if not entirely, estuarine. It ascends the river Hooghly, that's in India, at least as far as Calcutta, that is to say, for a distance of about 90 miles from the open sea, and can live in water that is practically fresh. So this is the reference for those of you interested. Now, when you look at this map above, which I took from Google Maps, this is a 20-kilometer scale, so you can think at first, well, that's pretty far from the coast. But actually, if you zoom out a little bit to the subcontinental scale, here you are. You're not so far from the coast. And if you look at it on a global scale, you are on the coast. What I mean by this is that, okay, these animals can venture into pretty low salinity water. One species can even venture into what is more or less fresh water, but never very far from the ocean. And this is very interesting because these horseshoe crabs in the Paleozoic have been found in localities where the inferred environment is not compatible, in my opinion, with this. For example, Montsolemin, where one species only occurs, but 142 specimens, well-preserved, three-dimensional, articulated specimens. So they are not carried far from where they live. And Montsolemin is most often interpreted as a freshwater intramountainous basin. Some even argued for a Tibetan-style altiplano, 3,000 or 4,000 meters above sea level. So I ask you, based on what we know of the extended distribution of Xiphocerans, is this a plausible interpretation of the habitat of Carboniferous Xiphocerans? I think not, personally. But, you have the right to disagree. For those who are interested, here is the paper by Bec Giraudon et al. In 1996 that suggested Carcinian high-altitude phenomena in the French Massif Central. So that's the paper that places Montsolemin fairly high. And the Rashkoff et al. Paper in 2002 describes beautifully the three-dimensionally preserved Xiphocerans from Montsolemin. This is one of them here. but there's many. Moving on now to another group of arthropods, also associated often with these stegocephalians, the Eurypterids. Fascinating taxa. In fact, they're fairly rare fossils, but in the Carboniferous, they're often associated with our predecessors, the stegocephalians. However, this association is much older. The first vertebrates lived alongside Eurypterids in the seas, and in fact, my predecessor, Alfred Sherwood Romer, a very famous American paleontologist from Harvard, even suggested that the first vertebrates acquired a thick dermal skeleton, some kind of carapace, as a means of defense against the fierce claws of the Eurypterids who tried to eat them, as you can see here to the right. Anyway, when our ancestors moved further inland, apparently the Eurypterids followed them. Maybe they wanted to keep eating them. So where did they live? Well, the first ones, notably those in the Silurian and early Devonian, were clearly marine. They're often found in marine limestone. But after the Devonian, there is less consensus. And in fact, for the Carboniferous localities, we see the same controversies as the habitat of the Xiphosaurans or horseshoe crabs. What I can tell you is that when they co-occur with stegocephalians, it is in the sites that have fairly strong marine influence, 4 sites that yielded these limbed vertebrates. So let's take the example of Adelophthalmus, which is reputedly one of the best adapted to freshwater, one of the scorpions that live the most inland in freshwater environments. It is present in localities like Joggins, that I already showed you, Mason Creek, Hamilton Quarry, El Cobre Canyon in New Mexico, the Red Tanks Formation, and many others. In many of these formations, it is clearly associated with a fauna that indicates marine influence, in my opinion. For example, you find cnidarians in Mason Creek, brachiopods in Hamilton Quarry and Joggins, not at all levels, but you do find them. Echinoderms in Joggins. Amphinomid annelids, these are typically marine annelids, which are often called polychaetes, but polychaeta is paraphyletic with respect to oligochaeta, so I will not use that term, but these are clearly marine annelids. Macroconchids, which we discussed earlier. So I think that in these localities, the sea was not far. Now, a few words about the so-called freshwater jellyfishes that are also recorded in many of the stegocephalian-bearing Carboniferous localities. First, the extant cnidarians are represented by more than 12,000 species. This is a very diverse taxon. Fewer than 35 of them live in freshwater. That's 0.3%. It's a minute minority. So how do we know that these jellyfishes or cnidarians that we find along with stegocephalians really inhabited freshwater environments? In the first approximation, we should consider that they are probably marine taxa, perhaps estuarine, but my first guess would not be that it's a freshwater taxa. And a recent study showed that the taxon that was interpreted as the freshwater medusa, or freshwater jellyfish, most common in Mason Creek, is actually a euryhaline sea anemone. Here are 2 pictures of the fossil to show you first, to the left, why it was interpreted initially as a jellyfish or medusa. You can see here, it seems fairly circular. So it was a good guess. But other fossils show that here you have the foot and here the mantle and the tentacles would be somewhere here. So this is a fairly extensive monograph about this taxon. They have all kinds of interrelated morphotypes too. So it makes a convincing case that the live animal looked like this. And they conclude that this was an estuarine euryhaline form. So it lived in brackish water, not fresh water. So there goes at least one freshwater medusa. We should know. Neither medusa nor freshwater. So now I covered more or less what I wanted to say about the associated fauna. We'll look very briefly at sedimentological arguments about the environment of these localities. And I would like to talk here only about tidal rhythms, which may be far more widespread than often believed. These formations are left by tides, and they leave the zoolaminations. And what allows you to determine that there are tidal deposits is that you can see evidence of neap spring cycles. So there are cyclical variations in depth, in the thickness of these layers, which reflect a 14-day periodicity, for example. So you have strong tides and you have little tides, and that changes along the month. And Kusharial in 1990 wrote that it is suggested that evidence of Westphalian tides may have been misinterpreted or overlooked in field studies elsewhere. So if you're interested in an early study that already warned us that we may have overlooked many tidal rhythms or tidalics, that's the same thing, this is a nice reference. And here you can see such deposits. Now, 33 years and about 24,000 tides later, we find this interesting quote by Dimakel et al. 2023. Such deposits are likely much more widespread than recognized because of the difficulty of identifying tidalics in their nascent, very finely laminated stages, particularly in mud rocks. However, the limited recognition of tidal flat settings also may reflect the fact that most paleobotanists are generally unfamiliar with these kinds of strata. And here they emphasize paleobotanists because the authors are paleobotanists. But you could say the same about vertebrate paleontologists, that I can assure you, and probably many other types of paleontologists. So sadly, in 33 years, we didn't make much progress in mapping out the distribution of these deposits apparently. I infer from this that marine influence may be more widespread than commonly acknowledged in Paleozoic sediments. Note, however, that this is not strictly a paleosalinity indicator because tides do spread inland into estuaries and rivers farther than brackish water. So they can reach into fresh water, and they do in some cases, but never very far from the coast, of course. When you see these, you can be sure that the sea was nearby. Let's now move to recent isotopic data. And before we do, I present you here a tree that I published in 2010 in my first review of this topic. What you see here is, in white, our finned primitively aquatic predecessors, taxa like Eusthenopteron, that people would call fishes, that they had paired fins, internal gills, lateral line systems. They were fully aquatic taxa. In white, what you see is those that live clearly in the seas, in salt water. In green, you see taxa that occurred in brackish water that were probably euryhaline. My new interpretation was already in 2010 that you had a shift from a marine environment to only brackish water fairly early in the Devonian, perhaps in the Gibetian, but that most Permo-Carboniferous taxa retain the ability to tolerate salt. You find here in this large evolutionary radiation, you find the Demospondals, Embolomeres, Simoiamorphs, Lepospondals, other taxa that I mentioned at the beginning of this talk. And in black, you have the taxa that are strictly freshwater, that do not tolerate salt. And notice here I arrowed two taxa in the Devonian. These are Ichthyostega and Acanthostega. Because in my review, despite having looked at the literature on these localities, I found no sure sign of marine influence. In fact, I had written an email to Jenny Clack, who's an authority in these taxa, and she assured me that this was a freshwater environment. She was pretty sure of it. Anyway, so I provisionally accepted this, but note that this implies a reversal from brackish water forms, or not a reversal, an acquisition of a strictly freshwater lifestyle from taxa that were euryhaline, a very early one, convergent with modern amphibians, which you find here today, various amphibians. However, it appears that here I was wrong in accepting this traditional view. This recent paper by Goddard, Lecuyer, Amiot, et al., published in 2018 in Nature, infers euryhaline ecology of early tetrapods. And this was revealed by stable isotopes. So they used mostly three types of isotopes. Here you have carbon, in which they showed that these taxa, well, which they call tetrapods, but they're not tetrapods in my sense, they're stegocephalians, they are stem tetrapods. But this includes Ichthyostega and Acanthostega from East Greenland. They overlap widely with extant sharks, which are mostly marine taxa. This is true also of placoderms and sarcopterogens you find in the same localities. This was a study of several localities from the Flammenian, Lake Devonian, including the one that yielded Sinostega, the Chinese form, which is very far. Here we have similar plots with oxygen and sulfur, other isotopes that have been commonly used to infer paleosalinity and paleoenvironments in general. The most interesting is this one here, which is a bit easier to interpret. This here represents seawater. So it's delta O18 and delta S34, so isotopic ratios of oxygen and sulfur, respectively. And here the little light blue dots represent different freshwater bodies, because freshwater, since it is in a continental environment, it reflects partly the isotopic composition of the sediments on which it flows, because minerals dissolve there, and so freshwater is much more variable in its isotopic signature than seawater, which is a very homogeneous medium because of the oceanic currents. Anyway, what you see here is that all of the fossils that they studied are somewhat intermediate between the various freshwater bodies and seawater. So they conclude that these inhabited coastal brackish water environments where you had a mixing of these two types of water, as you find in estuaries and deltas, for example. And this includes Ichthyostega and Acantostega from East Greenland. So Jenny Clack was apparently wrong on this, and I was wrong too in accepting her conclusions. So all of this leads me to conclude that the picture I proposed back in 2010 and even earlier, what you see here is taken from papers I published in 1998, is that whether you use the traditional phylogeny in which these amphibians are nested among the thermospondal, or the more recent and correct one, since it's mine, in which these amphibians are derived from lipospondals, the saltwater intolerance that you find in extant amphibians is a new character, an autapomorphy of this group. It does not represent, by any means, the ecology of the first amphibians or the first Stegocephalians. And this saltwater tolerance was probably lost to a large extent sometime during the Permian. But of course, it's not completely absent. A few years ago, when I went to Uruguay to collaborate with another colleague of mine, I saw frogs or toads that were reproducing very close on the beach in water where the high tide went. So these were obviously adapted to brackish water, and they even reproduced there. So some of the amphibians do tolerate some salt water to various extents. So what can we conclude from all this? Well, I think if we step back and look at animal life in general, it is pretty clear that life arose in the oceans and seas. And when you look at other taxa, like arthropods, it's clear that they originated in the seas in Cambrian, if not earlier. And you have then chelicerates that moved onto land in the form of arachnids, myriapods that moved onto land, insects, and so on. You see the same pattern in mollusks. The first mollusks were all marine, and today we have snails and slugs that are either terrestrial or freshwater. Annelids were also clearly marine in origin and moved into freshwater and the land later on. And I think for the vertebrates, it's the same thing. We came from the seas. Now let's move on to a different question, the origin and evolution of digits, because let's admit it, to move on land, digits are pretty handy. And so the tetrapod limb was originally thought to be an adaptation to a terrestrial environment. That seemed at first approach to be a good guess. However, this hypothesis was to a large extent refuted when we realized that some stem tetrapods that had digits, like perhaps in Herpeton, but at least Acantostega, where it's clear, had lepidotrachea in the tail, so they had a tail that is clearly a primitive tail for swimming in water. They have a lateral line organ, which is adapted to detecting motion in water, which you find in no secondarily aquatic tetrapod today. So this means probably a primitively aquatic lifestyle. And you had internal gills, which also occur in no extant tetrapod today. For example, the perenibranchiate urodales, like the acholodon, they have external gills, not internal ones. They're not the same. So now the idea is that the limb arose in an aquatic environment. And the question is, of course, why? And is this possible? Now, first, let me try to convince you that it is possible. If you look at arthropods today, the walking limb is almost universal. Of course, it's not surprising in a crab that is able to crawl onto land like this one. But consider that many arthropods, aquatic arthropods, have perfectly good walking legs and never had any terrestrial ancestors, like this crayfish and lobsters, for example. These are aquatic. In the Cambrian and Ordovician, you had many primitively aquatic arthropods. Most of them had perfectly good walking legs. So why would not our ancestors also have acquired walking legs while living in water? That begs the question, why? What did they do with these limbs? Well, one possibility is that they were used to anchor themselves onto the substrate to resist currents. It could be river currents, but it could also be a wave current, or the tides coming and going generates currents. It's used, for example, in the marine iguana to graze at the bottom of the ocean at shallow depths. Salamanders grasp onto stones in rivers. Frogs use them to copulate. The male holds onto the female. You could also imagine vertebrates moving into mangroves, which are very incomplex environments. It's not easy to swim because there's not a lot of space, but fingers can be very useful to grab onto roots and move along. So there's many reasons why the limb might have occurred in primitively aquatic vertebrates before they moved out onto land. So the next question is, when did the limb appear, and in which taxa? And here I want to draw your attention to an ambiguity in the literature. Many taxa that have been called tetrapods in the literature, especially the earliest one in the Franian and Flaminian, are neither tetrapods in the phylogenetic sense, because they are stem tetrapods, so stegocephalians, but not tetrapoda in the meaning I use, and they may not even have had digits. For example, a Guinea-proton. This is more or less what we know about Guinea-proton. We have a lower jaw, we have a bone that may be a humerus, although its identity has been questioned recently. It may not be a humerus, and it may not even belong to this taxon. And a tibia. Digits, we have none. And this is the case of many taxa. You see them arrowed in green, and they fit between the taxa that retain paired fins in white to the left, and the taxa that are known to have had digits. In dark blue here, you have the taxa that have polydactyl fins, that is more than 5 digits. This is our ancestral condition. At some point, we had polydactyl ancestors. And so it's no surprise that some babies, human babies, are still born with 6 fingers or 6 toes. It may be somewhat of an atavism, a return to an ancestral condition. Anyway, the pentadactyl taxa are here. So as you can see, the basal-most stegocephalians that have been called, not by me, tetrapods in the literature, even parsimony does not allow us to determine if they had limbs or fins. It could be either way. We'll never know until we find more complete fossils. So we can turn to ichnofossils to try to learn more about the origin of the limb and the timing of its appearance. The oldest potential trackway of stegocephalians has been described by Niewicki et al. In 2010 in Nature. This is from the Middle Devonian of Poland. And these are some of the best trackways that are preserved. However, it is so early, because it is Eifelian, that we wonder, is this really a stegocephalian? And in fact, it is not certain. Some experts in this field consider that it is probably not. And here you have 2 of the earliest papers that discuss this, Schulze in 2013, and Lucas, 2015. Both of these think that other interpretations are possible, and Lucas even thinks that it is definitely not a stegocephalian. But let's examine the possibility. If it is a stegocephalian, according to Niewicki et al., this implies several long ghost lineages in early stegocephalians, because they place it here in the phylogeny between Acanthosiga and Nyctaeciga. And that, I think, is a bit silly, because the anatomical information we have from these footprints is really not good enough to make this inference. And so this inference of several long ghost lineages that extend from the Eifelian to the late Flaminian is unjustified. The footprint could fit here into the phylogeny, and then this implies only one long ghost lineage, and the evolutionary radiation of the stegocephalians we know of can be much later. But it may not be a stegocephalian at all. And if it is not, then the next oldest potentials to diversify in trackways, which are much more convincing and probably are, they start in the Gibetian. So this is the late middle Devonian. Just a little more recent and still substantially older than Ichthyostega and Acanthostega from Greenland. Now, interestingly, Lucas in 2019, and he did a review on this topic, he claimed that these records were non-marine and that this did not lend support to a marine origin of our ancestors. However, this is inconsistent with his own map. Look at this. This is taken from his paper, Lucas 2019, figure 3. You have two occurrences here in New America, and you have one here. And look at all this sea in between. How could a freshwater taxon cross this? Seems difficult. And this is only Gibetian Freudian and Flaminian records. It does not include the later ones. So I think marine dispersals obviously occurred, according to the ichnofossils too. So now, last but not least, why did our ancestors come out of land onto land? Why did they go up? Did they leave the water? Several hypotheses have been proposed, and some are more likely than others. Let's view them in a random order. First, perhaps to escape predators. This is an illustration taken from one of my favorite books on prehistoric animals that I had when I was a kid. Because being a paleontologist for me was a childhood dream. Here you see Eusthenopteron, which was long viewed as our iconic ancestor from the Flaminian. It's known especially from Miguasha in my home province, Quebec, in eastern Canada. And here you see an adult one eating a small one. And the other wiser ones are closer to the edge of the water, in shallow water, where the bigger one wouldn't venture because it might get stranded. And one is starting to walk on land. This picture is probably wrong, but however, still today, you can see that many teleops, for example, when they're young and small and vulnerable, they remain in shallow water, where they are relatively sheltered from the numerous bigger aquatic predators that they might face otherwise. In lakes, for example, these peripheral ponds are very important for breeding the young teleops. So this is actually a fairly reasonable hypothesis. Maybe our ancestors were trying to get away from predators, because in the seas you had these terrible eurypterids, many of which were larger than our ancestors. You had large, fierce placoderms like this Dunkleosteus, which the head is like this big, 1 meter, just the head. It had terrible jaws. We had many sharks. You've seen jaws. Sharks can be terrifying. But on land, there were almost no predators, so it was a fairly safe environment. Now, another hypothesis that has been proposed a long time ago and now discarded is periodic drought. At some point, it was thought that our ancestors lived in freshwater bodies that became seasonally dry. And this was based partly on interpretations of the Texas red beds as arid deposits, and so the inference of seasonal drought. Now we know this is wrong, in fact. The red color does not require arid environment to form, and in fact it can occur, it can be acquired millions of years after the deposits are laid down. So there's no reason to think that our ancestors experienced drought, especially since, as I tried to convince you, they were probably marine. So this hypothesis has now been discarded. But if you look at old paleontology books, you'll see Eusthenopteron crawling out of a drying pond, trying to get to a deeper one. And that was a hypothesis as to why the land arose initially. Romer, notably, pushed this hypothesis among others. A more interesting, perhaps, hypothesis is for thermoregulation. Still today, you can see many reptiles, especially, basking in the sun. Now, if you wonder why they do this, is that one of the advantages is that when the sun is strong, they can raise their body temperature, and this accelerates their metabolism. This means they can be faster than their congeners that are cooler. Because you know, when reptiles are cool, they're slow. So when they go back into the water, they can outrun their prey or predators. So crocodiles do this, turtles do this. So why not our ancestors? Perhaps they came out to raise their metabolism. Was it for breathing air? Because there's more oxygen available in air than in water. But this hypothesis can be discarded because, as you can see, many teleosts, like carps, can come out at the surface of the water and got air. So you don't have to move onto land to breathe air. You just have to go to the water surface. So this one can be discarded. Was it to feed? Well, it might seem plausible today, but perhaps not back then, because there was not much to eat on land. There was very little animal life, although arthropods did move there earlier than vertebrates, so we cannot completely rule it out. There was, of course, abundant plant life, but we don't find so many herbivores, at least among vertebrates. We don't find any in the Devonian, not any in the early Carboniferous. They appear in the latest Carboniferous. So that may be not the most convincing hypothesis for vertebrates. Although perhaps they would have eaten some arthropods, but arthropods that were present then were not very appetizing, I would say. Another very interesting hypothesis is perhaps to lay eggs, because the land environments were less dangerous than the seas. Remember the first slide on this topic I showed you. There were many predators in the water. There were virtually none on land. And in fact, Romer suggested that amniotes became terrestrial because first they just went to the water, to the land to lay their eggs and lived most of their life in the water. His hypothesis was based on Limnocelis and Ophiacodon, and now we think that this may not be the best scenario, but it remains an interesting idea. Perhaps, indeed, some of our ancestors moved onto land to lay eggs in a sheltered environment. And this is something that you can still see today. Many frogs, especially in tropical areas, lay their eggs on leaves outside the water, or sometimes they keep the eggs on themselves. Many try to lay their eggs outside the water, even though there are now many more predators on land than there were back in the Devonian or Carboniferous. So it still seems somewhat advantageous today. This would have been even more true in the Devonian and Carboniferous. So, well, in reality, we cannot conclude firmly on this. This is a very elusive question, and I don't pretend I can give you an answer, probably not in my lifetime. But perhaps one day we'll know a little more about this topic. So to conclude, well, we can say that the land appeared before tetrapods, in the phylogenetic sense, probably in an aquatic environment. Early stegocephalians and their predecessors probably inhabited coastal areas, often in brackish water. And this is supported by recent isotopic data. The degree of marine influence in Paleozoic sediments probably remains underestimated, and notably many tidals, no doubt, remain to be discovered. I did not mention this at the time, but in some very intensively studied localities like Mason Creek, it took more than 30 years before it was noticed that they were there. So imagine in localities that are rarely studied. The digits probably appeared in aquatic taxa before vertebrates moved onto land. So this is not an adaptation to live on land, it's an exaptation, which some people call pre-adaptation. And we still don't know where vertebrates ventured onto land, but some hypotheses like egg-laying, predator escape, and thermoregulation seem more plausible than others, like drought, access to atmospheric oxygen, or possibly the search for food. And that's about all I can tell you today. So I thank you all for your attention, and for those of you who were interested in this talk, much of what I told you today is from a review paper that is currently in revision, so it passed the first filter of review, and I will resubmit it soon, called Habitat of Early Stegocephalians, a little saltier than most paleontologists like. The part on early land evolution is from my book, How Vertebrates Left the Water, published by University of California Press in 2010. And the portion on phylogenetic nomenclature comes from my book, The Advent of Phyllocode, published this year by CFC Press. Thank you. If there are Eurypterids, my guess is that the stegocephalians were probably not very far. And why do you think, how old is that locality? And how do you know this is freshwater? But here you have to be careful because most of the localities that I mentioned to you have been interpreted as freshwater initially, and many of them, I'm convinced, are not. You see, in the Paleozoic, we don't have, to my knowledge, sure indicators of a freshwater environment. Usually the conclusion that it's freshwater is because we lack clear marine indicators. But this is negative evidence. It's always weaker than positive evidence. So I don't know that locality, maybe it is freshwater, but you should look at it critically because a lot of premature statements have been drawn on these things. And if there are Eurocterids, my guess is that the sea was probably not very far.
¿Alguna pregunta? ¿Alguna otra pregunta? Cualquiera que, por más simple que sea. También entiende castellano.
Sí, puede preguntar en castellano.
Sí.
No sé si contestará en castellano, pero... Sí, sí, este tipo de huevo es una adaptación para la tierra. Some amniote eggs are laid in water sometimes, but there they remain in a dormant state and they start developing when they are emerged. So yeah, to my knowledge, no amniotic egg completes its development underwater. And the oldest one known is from South America, from Uruguay. It was described by my colleague Graciela Pinho in a paper that I co-authored with her in 2012, Historical Biology. It was soft-shelled, so the outer membrane was probably unmineralized, which is the primitive condition we can infer for the amniotic egg. See, when we talk about the egg, most people think about the chick egg, and they think, well, that's not representative of the early amniotic eggs, in fact. And in fact, this type of unmineralized or poorly mineralized outer shell membrane explains why the first amniotic eggs known, except for that mesozoa, which was soft-shelled, they only from the Triassic, whereas amniotes appeared in the Carboniferous. But soft-shelled eggs are unlikely to be preserved. You have to have the embryo. It's the embryo that we notice, not the shell, of course, because the soft shell has a very low fossilization potential. You're welcome.
¿Podría ser que algunos heterocefalians se hayan originado en el ambiente marino, pero posteriormente se hayan adaptado a ambientes de agua dulce?
Of course, I think that this occurred. The question is, in which taxa exactly and when? Yeah, it has the traditional position about this is that even in the Devonian, taxa like Ichthyostega and Acanthostega were freshwater taxa, right? Which would imply, since these are primitively aquatic, that our ancestors moved from the seas to freshwater and then onto land. And I think this is wrong. I think they moved from the seas onto land and perhaps freshwater at the same time. And perhaps some of them that lived into land went back into freshwater, because this occurred multiple times in evolution. You have freshwater turtles, freshwater otters, freshwater frogs, of course, and many others. So what I'm trying to say is that I think we cannot, it's dangerous to generalize. I think that many of these taxa that were assumed to be freshwater, many of them were not necessarily freshwater. But of course, I'm not saying that none lived in freshwater. There must have been some. But it's more difficult to mention, to know which ones, because as I answered to the guy behind you, we don't have clear freshwater indicators. We can conclude that it was probably freshwater if we have no marine indicator and no salt indicator. Because here we have to be careful not to confuse freshwater and continental water. Because a lot of continental water is fairly salty, right? Great Salt Lake, Dead Sea, and many others. And you have brackish and salt lakes in every continent, and not only in arid environments. We have them in Europe too. And in the Permian of Germany, there was a large perennial salt lake. So continental does not equal freshwater, even though this has been kind of assumed in much of the literature on the topic. If they found no marine fossils, they conclude it's freshwater. But look at the Aral Sea, Caspian Sea, Black Sea. This is not a truly marine environment, but it's not freshwater either. There are many intermediate environments, and relatively little has been written about this, and very few of the localities have been interpreted as such. And I think this is anomalous, because if you look at where the sediments get laid down today, we have a sedimentologist here, so I think he can tell us more about this, but most of them end up in the seas, or at least in large perennial water bodies, in deltas and estuaries, not somewhere between the mountains and the coast, right? Because some gets deposited in between, but after that, that's reworked and it ends up in the seas. So I would expect most of the late Paleozoic sediments we have to be coastal deposits. And this is what suggests, the evidence that I read would suggest. There were probably freshwater environments that were inhabited by stegocephalians, but I fear that we probably know these much more poorly because they have been likely to have been eroded in the meantime.
¿Alguna otra pregunta? Yo puedo continuar con la que hizo él. O sea que yo tengo una pregunta debajo de Belis, o sea que puede ser que es probable que encontremos que haya habido un mar cerca en esa época para los geólogos.
Tengo entendido que Bajo de Beni formaba parte de lo que era la cuenca de Paganzo,
Sí, sí.
pero era netamente continental. Si bien la cuenca de Paganzo estaba conectada a la cuenca de las iglesias, que era marino continental, porque tenía ingresos y egresos del mar. Pero la cuenca de Paganzo en sí era netamente continental.
¿A dónde estaba el borde de contacto con el mar?
Al oeste.
Sí, 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 pero más o menos a qué distancia, ¿sabes? Porque si la mayoría de los eudictéridos eran marinos,
Al principio es claro,
¿hasta qué periodo?
hasta el Devon más o menos.
Habría que ver si realmente los autores que dijeron que la araña, Megarachnis sardines, es un eudicterio, habría que volver a revisar si realmente es un eudicterio o es una araña, porque si no, los eudicterios hasta el devónico eran la mayoría marinos. Es como que se necesita un poco.
This discussion raises two important points that I'd like to comment on. In fact, Hans-Peter Schulze had already commented in papers published back in the 1990s that it's dangerous to use reconstructions of paleo coastlines to infer the habitat of a locality because it's somewhat circular, because these were based on previous interpretations of paleo habitat. And these, yes, and these may have to be revised in some cases. For example, there was a large monograph on the development of freshwater ecosystems published back in the 1980s. It's referenced in my paper, but I don't have the author's name in mind now. That wrote some very rude and harsh comments about Hans-Peter Schulze's work about this, saying that he ignored a large body of data and how ridiculous is it. The sea was hundreds of kilometers away. She was talking about the Escamilac Formation, which we now know is a estuarine. Isotopic data proved that a few years later in the 1990s, and now it's almost universally accepted. So the paleo coastlines were not drawn correctly. Also, second point related to this one is that paleo coastlines, of course, change with time. For example, the Black, Caspian, and Aral seas are remnants of the Parathetis, which was a marine environment. But oceanic connections were cut. After that, the typically marine faunal elements were progressively lost over time. Not immediately. I collaborated with some paleomammalogists and we described some whales and seals from these environments in the Miocene, Pliocene. So in the past, there were surely such water bodies that had been connected at some point to the seas and had lost this connection. And perhaps Euroctid is in one such environment. Perhaps at that time there was no connection to the sea, but perhaps there had been 10, 20, 30 million years earlier. And perhaps these marine-like faunal elements are remnants of this. In the Black Sea today, of course, which remains connected indirectly through the Bosphorus with the Atlantic Ocean through the Mediterranean, you still find a few marine elements, but very few. I know because I swam there in the Crimea, where I wouldn't go right now. I don't recommend. Question.
Sí. ¿Han evaluado forzantes climáticos en cuanto a la aparición de los apéndices?
Which climatic aspects?
Sí, como glaciaciones,
La-
glaciations.
Well, I didn't look at that closely. What has caught my attention, though, I mean, it may be interesting to look at, but I haven't done it. Maybe this is something you should look into. But the regressions and transgressions may have had an influence. This is an idea that Halcon Lang and his collaborators have pushed since the early 2000s. And they argued that in the late Carboniferous, there had been many transgressions and regressions, and that this left many epicontinental seas and some perhaps brackish water environments, and that it may have favored the appearance of many brackish water forms, Euryhaline taxa. So this may have been an important step in the development of freshwater ecosystems, because this was probably something gradual and probably a repetitive pattern. Glaciations, yeah, they're probably connected with this too, but I don't recall seeing this point being made. It should be investigated. It's a good idea.
Bueno, si no hay más preguntas, agradecemos al doctor.
Gracias. Gracias.
No, me fijé. Digo, voy a ver cómo está saliendo. Resulta que estaba la pantalla olvidada. No. Y que no sé cuáles son. ¿Viste?
Sí. Sí.